One of the things I want you to pay attention to, and this kind of sounds whining, but I'm just going to say it, almost every word that comes out of my mouth is important. I record and re-record these because I say one word wrong or not the way I think it comes across best. So pay attention. And if you don't understand something, look it up. Use your book. Go to the internet and explore. Coding regions of messenger RNA retained are called this. After a messenger RNA is formed from DNA in the nucleus, it is carried out to the cytoplasm where it forms a protein in the process of called translation on this organelle, by the way. But what's kind of interesting is that the original copy of messenger RNA is modified. There are non-coding parts which are removed, and that's what these are called. I used a lot of artistic license here, so bear with me. I don't think I've ever seen anyone do a drawing exactly like this, but here goes. When a messenger RNA strand arrives at the ribosome, which is a different kind of RNA, by the way, it's ribosomal RNA, 3 bases make a codon, and that tells another kind of RNA, transfer RNA, which amino acid to bring over to assemble. This is how messenger RNA directs translational, how would I say this? Air traffic control? You know, how to tell which airplane to fly in next, which shuttle to bring in the next amino acid, and what the order should be. Notice that I have 3 prongs on the end of this little arrow. This is kind of representing my transfer RNA, which is shuttling the amino acid, and this 3-pronged interface would be the anticodon on the transfer RNA, which matches to the codon on the messenger RNA.